Yeah, I brought it.
We'll start in like 3 minutes and see
Yes,
everybody
has
have
speakers.
please help yourself there. The sodas, the burritos, from those.
That's what from the announcement I just said we'll be teleconnected with David.
David, like that again,
General writing, just saying
like where in David? Yeah. Yeah, it's not clear unless you've been here. Once you've been here, and then like we're good.
That's what I thought.
Although I still don't know what part of health restrictions and stuff. Yeah, I'm in a little office building on the boulevard, and like the parking always confuses me.
Yeah,
He told me at a certain lots. I called him and said certain lots.
Yeah, I'm so happy today because somebody called me out there from Santa Clara University, and they called and wanted to talk about that. A non-Zika question, yeah. Should be doing like one thing a day. So for today, But they can put on the suits outside, right? Yeah, I kind of wonder. Hopefully it's not like a comedy show or Well, you can see that. Like on 60 Minutes, how sometimes it's like if they put on the suits, it would make them look as if they've got more access, like they're really getting the inside story, right? So,
Either that or they were truly worried.
or they were worried, yeah. Yeah. It was just like weird genetically modified mosquitoes or something.
Regulations, you have to get
Right. Like more than in China.
Let's start. First of all, welcome. This for those of you that are new faces that haven't seen here before, this happens on the first Thursday of every month, usually in this room, sometimes on the Davis campus, and deals with all things global health considered. And we're completely open to ideas. The agenda isn't a political one, except to bring new ideas, new thoughts, new concepts, new experiences to the medical, nursing, veterinary community. So if you've got ideas for topics, please let us know. We'd love to hear them. Tonight, we've got two distinguished guests who are going to help us put in context what we read about all the time, which is the Zika virus. It is the newborns with these misshapen heads that seem to dot the pictures of the New York Times, the Wall Street Journal, and the LA Times, and what have you. And they're scary and they're horrible and they cause lots of fear around the world and lots of response from the CDC and from WHO and from other organizations. There are reportedly so far thousands of babies born with microcephaly in Central and South America, and potentially many more to come. The problem, as we'll hear, is that the phenotype, the condition of microcephaly, has lots of things that lead to this. And so it's sort of hard to study, and lots of theories and lots of worries. Microcephaly means a life of uncertainty for the child, but also for the parents. It ranges from really 10% of kids who have microcephaly have no symptoms at all to kids who have profound disabilities. I'm sure we'll hear about that. Microcephaly is a clinical diagnosis. It's made no sooner than 24 weeks during pregnancy. At least in North America, most women get an ultrasound at about 20 weeks of pregnancy, meaning that it isn't detectable at that point. Most women will end up going and delivering a child and only finding out at birth or shortly after birth that they have the condition. It also should be pointed out in the United States, we have 25,000 births with microcephaly every year. Presumably none of those, unless we hear otherwise, none of those are associated with the Zika virus. So this is something that happens aside from Zika. We may hear in a short time from Dr. Blumberg that that microcephaly itself is associated with lots of viral conditions. It's associated with diabetes and alcoholism and Down syndrome, cytomegalovirus, and toxoplasmosis as well. So it is, at the end path, you can get there several different ways, and we need to understand about this virus, both in terms of its implications on overall global health, implications on local health. As you may know, mosquitoes are a big problem here in California, not Zika, and perhaps not this type of mosquito, but we're eager to hear if this may be something that we find here in California. So we've got two experts. One of them is a vector disease biologist, Chris Barker, Barker. I apologize for the misspelling some of the notices. And Chris will give you his bio. It's been on notices that you've got, but he spends lots of time thinking about little teeny things that spread diseases to often much bigger things. Dean Blumberg is a pediatric infectious disease specialist, and he spends lots of time thinking about little teeny things that cause disease and bigger things than humans, particularly, and mostly in pediatric age groups. So we're delighted to have them both. I think perhaps with the cost of recording, although I wasn't there, the decision was that Dr. Blumberg would go first. So good. Thank you both so much for coming.
Great, thank you.
So with the systems of being from CDC slide, no audio. Well, I usually do like to show a lot of audio because then I get to watch and take a break, which is always kind of fun, But I do my disclosure slide, which I am on Speakers Bureau for some vaccine manufacturers, and I always put this discussion on that I may talk about off-label use, although it's hard for me to imagine, but every once in a while I start talking about off-label use stuff. So first I'm going to talk about transmission just briefly. I mean, the major transmission we've all heard, of course, of Zika virus is mosquito bites. But there is other transmission. Just a couple of days ago, we heard in the news of sexual transmission of Zika virus. I'll talk briefly about that. Has been reported with blood transfusion. You might have heard of some new recommendations regarding blood transfusion. That anybody who's been in the Zika endemic area now should not donate for 28 days after their return. And laboratory exposure. There have been some transmission also. And then there's theoretical transmission, which hasn't been documented with organ transplantation and breast cancer. But really, the vast majority of transmission is mosquito bites. And in terms of talking about the clinical issues related to Zika, I think this is, a nice article to start with. And Chris will talk more about the epidemiology and how it got to Yap Island. But there was this outbreak in Micronesia in 2007, and CDC and other investigators went there, and they were able, in this relatively close community, to do some really nice studies of epidemiology and clinical characteristics of Zika virus. And so I wanted to show some information from that. First, I figure not everybody knows where Yap Island is. I didn't. So, of course, Australia is here, and then there's Papua New Guinea and Indonesia, and then there's this little island up there, Yap Island. And so it's really kind of isolated from other areas, and they were able to get some good studies from there. They found the infection rate in this naive population was 73%, so quite high. But the symptomatic attack rate was pretty low, so only 1 in 5 people who got infected actually had any kind of clinical symptoms. All age groups were affected, but adults were more likely to present for medical care compared to children. And there was no severe disease, hospitalizations, or deaths. However, there's a case report of a patient with sickle cell who's going to be published, not yet published, who did die reportedly of Zika virus. So you can't have deaths from Zika virus. This is the age distribution and the attack rate in that outbreak. You can see the confirmed cases in the blue, and the white boxes are probable cases. It looks like there's a higher attack rate among younger and older adults compared to children. These are the clinical characteristics of the patients with symptomatic Zika virus. You can see 90% had a rash. 2/3 of fever, arthritis, or arthralgia, and then non-arterial conjunctivitis, and then a variety of really non-specific clinical signs reported. But I do want to highlight these 4, because this will come up later with some of the CDC guidance, and these are the 4 most common manifestations. And I guess they didn't include myalgia because, arthritis, arthralgia, myalgia, whatever, it aches, right? So it's kind of included there. And then I just wanted to show a picture of the rash. So I took this from the European CDC publication, which published a couple cases of rash and conjunctivitis, the non-purulent conjunctivitis, just irritation of the conjunctiva. Nothing specific about this, really. You can get this from a lot of infections. The rash, this is the back on the front of a patient. This non-specific macular papular rash. Nothing really specific about that. We see this in kids all the time related to a lot of community-acquired viral infections. There's nothing specific about it, like, for example, that we see with measles, which is a more characteristic rash, or chickenpox, for example. The clinical course is generally mild. Symptoms last for 3 days, up to a week, and severe disease is really uncommon. Fatalities are rare. One thing that's been associated is in Guillain-Barré syndrome. Reported the relationship is unclear. I'll talk about that in a moment too. But the major issue has been this toxicity that's been reported with this recent outbreak in Brazil. And what they found is a substantial increase in newborns in 2015 in Brazil, but they don't know about the true baseline. They have some data on baseline data. So that's why you'll see that a lot of people aren't saying that it's positive, they're saying it's an association. And Zika virus infection has been identified in infants with microcephaly, infants who've died, and in some miscarriages also. But some patients, some kids born with microcephaly have been negative for Zika virus. So not all cases are associated. And then the incidence of microcephaly with congenital Zika infection is really not known. So we have really more questions than answers right now. This is a picture from the AP News Service of a child with microcephaly. Their heads are small. And the reason that their heads are small is not because the skull restricts growth, but it's because the virus actually attacks the neurons, the brain cells, and it kills them. And so the skull doesn't grow normally because the brain's not growing normally. The European CDC was a little bit ahead of the CDC on this. This was from December, where they asked for a risk assessment of the association with microcephaly and Zika virus. And what they looked at was several different states in Brazil and looked at the baseline number of reported cases of microcephaly, the total being about 150 a year for the 5 years preceding 2015. And then you can see the large increase in reported cases of microcephaly that occurred. And then if you look at this by state, the top 5 states, I just want to call your attention to this because this will come up in a moment. Really the top 5 are the vast majority of cases of microcephaly in Brazil. And when they look at those top 5, you can see they can look at the average number of births in those states, the number of cases of microcephaly before Zika hit in 2015, do the same after, and then get a ratio here, the ratio between the increase in 2015 compared to the previous 5 years. And you can see in some of these states more than 70 times the number of cases of microcephaly that we're seeing. And again, the top 5 here really quite stand out. So I'm not a whiz with geography related to Brazilian states, but I did take a look, and these 5 states are right here. These are the 5 states, including the one in white, where they say that no Zika has been reported, but clearly it has a big spike in microcephaly cases. So you can see these are really all grouped in the northeast of Brazil. This is from the CDC. It's a similar analysis, but it's updated by a few weeks. Each dot represents, the size of the dot represents the microcephaly rates in each state. And that's in the 5 years preceding the outbreak. And then this is 2015. And again, this large number of increase really concentrated in the northeast of Brazil. So microcephaly just means a small head. And there are standards for microcephaly, so there are degrees. You can have a small head because your parents have a small head, and your brain can be completely 100% normal, have normal neurologic outcome, normal development. But a small head is a reliable assessment of the intracranial brain volume. So you can imagine if it's really small, that means there's a lot less brain. And this can lead to cognitive sequelae, developmental and neurologic sequelae. So children who are born with, who are severely affected with microcephaly, they can be mildly affected, they can have seizure disorders, they can have motor development problems, fine motor problems, they might be deaf, they might be blind, they might be so severely affected that they die because they can't even eat normally and they aspirate. So there's a whole wide range of outcomes with children with microcephaly. And of course, that's the major concern now in Brazil, is not only it's a tragedy for the individuals who are affected, the individual children who are affected, and their families, it's a tragedy for them, but you can imagine on a public health scale, if you have a large increase in cases of microcephaly, that's going to be a big burden on the healthcare system, especially in a developing country, and that healthcare system is going to have to take care of those children. That's going to divert resources away from others. So it's both on an individual and public health level. Now, the mechanism of microcephaly, again, is the brain doesn't grow, so the head doesn't grow. There's a lot of primary, usually genetic causes of this, and then there's secondary causes. So the secondary causes is something happens that causes the arrest of the normally developing brain tissue. And a lot of these are infections. There's several infections that Dr. Wilkes alluded to that can cause arrest of brain development in utero. So cytomegalovirus, rubella, toxoplasmosis, these are the sort of things that when we see patients with small heads, when they're born, the neonatologists and pediatricians know to do a workup for that. So they might start with a genetic workup. They might start thinking about other associated signs and symptoms and start looking about other infections that might cause that. So what we know is that there have been a small number of infants with microcephaly who have tested positive for Zika virus. But what we don't know is the exact causal relationship between Zika and microcephaly. So we know that most of the cases of infections that result in microcephaly these happen because infection usually occurs in the first trimester of pregnancy. But we don't have documentation during this outbreak of when infection is occurring in pregnancy, so we don't know about the timing. And then we don't know, like, if a mom is symptomatic or asymptomatic, does that matter in terms of her child being more affected or not? Since 80% of the infections are asymptomatic, does that mean those 80% don't have to worry or not? Because we know from other viral infections, for example, CMV, cytomegalovirus is often asymptomatic when patients get that, and yet the child can be severely affected. So again, in fact, the severity of maternal infection, and then the magnitude of the risk and other adverse pregnancy outcomes, because we can't give any advice right now. We can't say, oh, you have Zika infection, and so the risk is 2%, 10%, 20%. Don't know. We don't really have that kind of data yet. So that's the kind of data that's urgently needed during this outbreak. And then the other thing that I think is maybe even most important is the possibility of cofactors with microcephaly. Because there have been outbreaks of Zika virus in Indonesia, and there was a couple suggestions of microcephaly in those outbreaks, but not to the same magnitude as has been occurring in Brazil. So are there nutritional factors that might make it be a cofactor? Are there genetic factors? Could there be two viruses? So could there be co-infections? Because that's what's needed to cause microcephaly. So there's still a lot of questions with this association. So there's limited information available about Zika virus in pregnancy, but there is no apparent increased susceptibility among pregnant women. An infection can occur at any trimester, incidence not known, but there is no evidence of more severe disease during pregnancy. So in terms of maternal fetal transmission, infection has been confirmed in infants with microcephaly and in infants delivered in the U.S. whose mothers have traveled to Brazil but then delivered here, so clearly they didn't acquire it here. The Zika virus RNA has been identified also in miscarriages and been detected prenatally in amniotic fluid. So a woman might have an amniocentesis. In two women, it was detected in 30 weeks gestation with a history of compatible symptoms, fever and rash of Zika virus infection. And then the children did have microcephaly and calcifications on ultrasound. So, it really seems like a strong association. In the Polynesian outbreak, there was evidence of fetal perinatal transmission. During this study, they had two pregnant women who had illnesses consistent with Zika infection at the time of delivery. The women had positive PCRs for Zika virus, and then Zika was confirmed in the neonates, one at 1 day of life, the other at 3 days of life. These children had positive PCRs, and it's really unlikely that the kids got it from mosquitoes between 1 and 3 days of life, so they probably got it from their mother. So this certainly is plausible. And then I also just briefly want to mention something that's had a little less attention, Guillain-Barré syndrome, which is it can be mild, but it's paralysis. And so paralysis obviously is scary, recovery is uncertain. In the Polynesian outbreak, there was a report of Guillain-Barré syndrome appearing within 1 week of confirmed Zika virus infection. In Brazil, there's really sporadic reports of an increase in Guillain-Barré syndrome. But again, what we suffer from is a lack of baseline data. So people just start looking for it, they can see it, but maybe it was occurring all along, we don't know. So again, more data is needed. In terms of diagnostic testing for Zika virus, PCR can be done in serum in the first week of illness. Serology can be done also. There's IgM for neutralizing antibodies. There's a plaque reduction neutralization test. This is a little tricky and has some cross-reactivity to it. And there's limited labs that test for this. So the CDC, as far as I'm aware, is the only one who's testing for this now. I don't think that CDPH has testing available. And then you usually don't get tissue, but if you happen to have a biopsy of tissue, you could also get do staining for antigens for PCR on tissue. So patients who you suspect PCR, there is no specific antiviral therapy. Treatment is supported, rest, fluids, give analgesics so they feel better, antipyretics to bring fever down. And then if you have suspected Zika virus infection, you should also evaluate them for possible dengue or chikungunya virus infection, because the same vector can transmit those viruses and the geographic area overlaps. And with dengue, you wouldn't want to give aspirin or other non-steroidals because you could increase risk of hemorrhage. So that's another reason to evaluate them for these other infections. And then in terms of clinical surveillance, the CDC has some guidance for that. They say, really, for clinicians, be on the lookout for patients who have acute onset of these 4 things: rash, fever, rash, arthralgia, conjunctivitis within 2 weeks after return. And then they say women who are pregnant, who travel to Zika endemic area, inform and evaluate, even if they didn't have symptoms, because most infections are asymptomatic. And then fetuses and infants, women who are infected during pregnancy, evaluate them for possible infection and sequelae, including microcephaly. And then, of course, report suspected cases, because we need to see if there's any cases. So prevention, there is no vaccine, there's no medication to prevent it. Primary prevention, reduce mosquito exposure. So that's why we've all heard the CDC recommending pregnant women should consider postponing visits to Zika endemic areas. But, they make these recommendations, and then as a clinician, I get questions. And so the questions I get are, I'm pregnant, and I know I shouldn't travel, but my father's dying, and he's in Brazil. So, am I going to tell her not to go be with her dying father? I mean, that's a very personal decision. So, I think you have to individualize it, and you could go and do mosquito bite prevention measures. So these are common that we're familiar with. Insect repellents, with any of the routinely recommended repellents, deep piperite and oil of lemon eucalyptus. Decrease the area of skin that's exposed to for mosquitoes to bite. You can do permethrin-treated clothing. A lot of people don't like that because it can hurt the clothes. Stay in a screened-in, air-conditioned rooms where mosquitoes can't get in. And then the CDC also has guidelines for pregnant women in the U.S. I'm not going to go through the whole algorithm, but it goes through whether a woman was symptomatic or asymptomatic, if she was in a Zika endemic area, an extra screening where you might want to do diagnosis of her to see if she's infected, and maybe do more ultrasound. And then there are recommendations for the newborn, also if the mom was in a Zika endemic area during pregnancy. And most of these relate to evaluate the child for sequelae, including if they have any intracranial calcifications or microcephaly, and additional diagnostics, and then neurodevelopmental follow-up. And then even kids who don't have any sequelae, there's still increased recommendations for surveillance. I do want to just come back briefly to the sexual transmission, because it was a big deal in the news on Tuesday? I think it was Tuesday it hit the news, the case in Dallas that there was sexual transmission. This is known. There were a couple cases reported from an outbreak, well, not from an outbreak, from there's endemic transmission in Senegal. 2 research scientists returned. They were married, they had intercourse with their wives, and then their wives who did not travel to Zika endemic areas ended up with rashes. And being good research scientists, one of them took a picture of his wife and her rash, and again, you can see on her back this non-specific maculopapular rash. So there'd be no way for them to get Zika virus. So a few years later, when a man presented for evaluation after being in a Zika endemic area, and he actually had hematospermia, which I hadn't heard of that term, but blood in the sperm. The scientists got the idea, maybe we should look in the sperm to see if we can detect Zika virus, because we know it can be sexually transmitted, and sure enough, it was detected in the semen. So this is certainly plausible. So I'd just like to end with some questions. We need to know some more information about Zika. What is the incidence of maternal fetal transmission by trimester, and what are the other cofactors, influential factors? What is the risk of microcephaly and other neonatal outcomes, such as miscarriages? And what is the risk of Guillain-Barré syndrome? Is this a true association or not? And so just to summarize, currently it's a disease of travelers, the clinical illness, generally mild, fever, rash, arthralgias, conjunctivitis, maybe an association with Guillain-Barré syndrome, and then the association with microcephaly, so that pregnant women should consider postponing travel to the endemic areas to decrease risk to disease. That's all I was going to talk about the clinical aspects. Chris, you want to?
Sure.
Then maybe we'll do questions afterwards and discussion afterwards.
All right, so as you might guess by the pictures, I'm someone who thinks more about mosquitoes than the virus much of the time, but we do dabble in the virus. My colleague, like Coffee, in my Department of Pathology, Microbiology, and Immunology is doing a lot of work on virology and the evolution of viruses that mosquitoes transmit. Let me just do it this way. Sorry, just 1 second.
There you go.
All right, so you might recognize. I just wanted to point out that there's lots of misinformation about Zika. You might notice this is straight from your UCD Health System website. You go to Dr. Bloomberg talking on YouTube, and then down here it says, are Bill Gates' vaccines causing the Zika virus outbreak in Brazil? If you click on that, there's some pretty interesting material, actually. And then there's also bioweapons. Zika virus is being spread by GMO mosquitoes funded by Gates. Well, GMO mosquitoes are being funded by Gates. There's actually a small town in Brazil that's actually south of most of the Zika activity that has mosquitoes being released. This is very small scale right now by Oxitec, who a lot of you have probably heard about in the media. They've also been trying to do some work in the U.S. as well, releasing mosquitoes with lethal alleles, actually, that where the males mate with the wild females, they don't produce viable offspring, and that's a way of controlling the mosquito population. So that's been translated by some in the pseudo media to mean that Bill Gates has caused the Zika outbreak. So we're here to set the record straight on some of this stuff. So what is Zika virus? I think by now you've heard most of this. It's a flavivirus. So flaviviruses are notoriously complicated. Dengue being a flavivirus, there's 4 serotypes of dengue, so essentially 4 different viruses that are dengue. So serology in particular gets really complicated when you get sequential infections of the serotypes of dengue. Zika just adds another one to that list. So you would have reactivity to dengue if you've had a Zika infection and vice versa. Aedes aegypti and Aedes albopictus are the competent vectors, Aedes aegypti being number 1 as a vector of dengue, chikungunya, and Zika worldwide. It was first isolated from a rhesus monkey in the Zika forest of Uganda in 1947. I'll talk just briefly about that a little more. And then first isolated from humans in 1952 in Uganda and Tanzania. And then in East Africa, there's thought to be a sylvatic cycle where it involves non-human primates and a variety of sylvatic, meaning forest-dwelling Aedes mosquitoes, and then also some paradomestic ones, meaning ones that are found around houses as well. So this is where the Zika forest is. This is Lake Victoria, which straddles Kenya, Tanzania, and Uganda. One Health groups on our campus are actively doing research there, as well as others. There's plague studies by CDC in Uganda. But this Zika forest is right along the border of Lake Victoria, and in some of the glory days of arbovirology, when people were discovering lots of new viruses in the 40s and 50s, this was a place where they were actually sampling monkeys. They put out sentinel monkeys. They actually were monitoring the monkeys' temperatures daily for fever, and then one of the monkeys was febrile, so they actually took a blood draw from that monkey and injected it into another monkey. The next monkey didn't show any symptoms of the virus. The first monkey, of course, had fever, but then they could show serological reactivity to the filterable agent that they got out of the first monkey. So with that, Zika was discovered in this Zika forest. And at that time, pretty interesting. There's local mosquitoes that they eventually isolated it from as well. So clearly there was some virus circulating between monkeys and mosquitoes. As I mentioned, eventually isolated from humans, and then this is what we were looking at for the Zika cycle. It was mostly a footnote in medical entomology classes, for example, until recently, as you just heard. So we got this human, mosquito, human cycle in urban areas. We have this sylvatic cycle, and if you sort of insert yellow fever instead of Zika, this is a classical sort of yellow fever scheme too, where there's a forest cycle between forest-dwelling mosquito species as well as non-human primates. Sometimes humans get infected in those cycles and then can initiate urban cycles as well. There are places to intervene in this cycle. Again, vector control with reducing the mosquitoes, which reduces the biting and the transmission, as well as the personal protection that Dr. Blumberg mentioned. Just mentioning more recent outbreaks, there wasn't actually a lot of work or even attention paid to Zika for about 50 years or so. And then in 2007, that's the outbreak Dr. Blumberg mentioned on the island of Yap. That's in the Western Pacific. He showed you where it is. 185 suspect cases reported, and the fact that 3/4 of the people on the island were infected is amazing, because herd immunity in humans eventually dampens transmission. So to get 3/4 of the people infected, you got to be pretty explosive in terms of the amount of transmission on that one island. This Aedes henselii actually, is the mosquito species that was incriminated there. It's in the same subgenus as Aedes aegypti, and that's also true of some of the sylvatic species in Africa. So the Stegomyia subgenus has all of these different species in it that are all related to Aedes aegypti, and in almost every case, if not every case globally, Aedes stegomyia species have been the incriminated vectors. In Asia, Aedes aegypti is considered an important vector. The virus has been identified in those species, and then also they've done, in Singapore, for example, there's been work done to show that Aedes aegypti can transmit Zika. So there has been some lab work, although not as much as we'd like. Then in October 2013 and 2014, we had the outbreak in French Polynesia, which shifts the whole thing out into the middle of the Pacific Ocean. 8,750 cases were reported. Some of us started getting interested at that point, but again, it's still associated with mild illness. It's in the tropics, you got so many causes of mild febrile illness that, well, okay, it's just another one. So there's bigger problems to deal with. In 2014, cases from New Caledonia and the Cook Islands, and then some secondary autochthonous, meaning local transmission, reported from Easter Island off the coast of Chile. So at that point, the problem seemed to be continuing in the Pacific. I think I'll skip most of this because I think you showed most of this. But seasonality-wise, it's sort of consistent with some of our other mosquito-borne diseases. Clearly, strong seasonality limited to the warm parts of the year. You showed most of this. Age distribution, a little bit older ages were affected most in terms of the symptoms, at least, which is also consistent with some of our other mosquito-borne viruses. And then there's this increase in microcephaly in Brazil. You just heard a lot about that. So really, it's thought to have come in with the World Cup potentially in 2014. There was also an international canoe race, and some media reports I've seen some people suggesting, well, it could have been that as well. Some large influx of people from Zika endemic countries, and particularly the closest viral isolate I'll show you at the end is from French Polynesia. So in terms of the phylogeny, that's the nice thing about viruses. They're relatively simple in terms of their genetics, and then viral sequences allow you to at least roughly connect the dots to the extent that you have viral isolates. So it seems probably that someone from the Pacific Islands, French Polynesia perhaps, arrived in Brazil probably in late 2014, and then by early 2015, public health officials there are starting to notice something's unusual here and start seeing this rise in microcephaly cases, as well as Zika itself. This is just another map from the media on microcephaly cases. So just as Dr. Bloomberg said, concentrated around Recife, which is right here in the corner and in the northeastern part of Brazil. And then now that's raising a lot of questions about the Olympics, which will be down here. There are football stadiums across parts of Brazil that don't quite get up there, but still lots of concerns about whether they should be canceling the Olympics. Just today, people are calling from the media, at least, wanting to know, is this a reasonable thing to do? Which, for Brazil, already dealing with a lot of economic issues, that's not ideal to be canceling the Olympics. I'll skip this. This is just the states that you've already seen with local Zika cases. Skip that as well. So this is what we're looking at now, sort of spread across the South America. We're doing work on dengue here with Tom Scott and in Iquitos, Peru, and that dengue is consistently transmitted down there every year. This is the rainforest side of Peru, and the Amazon sort of continues from Peru into Brazil. But we have not seen it in Iquitos yet. Everyone's sort of bracing for it and waiting, and it will complicate our dengue work for sure. But then we've also seen cases in Mexico recently, mostly in this part of Mexico, a little bit far from our border, but that brings it a little closer to us and also a little more in line with our seasons, because one thing we're benefiting from right now is that the southern hemisphere summers are our winter. So if imported cases arrive now, it's our low season for mosquitoes, it's our low season for temperature, and transmission doesn't happen very efficiently in those conditions. So one thing I wanted to highlight is what we're doing about this in California. My group has a pretty strong collaboration with the California Department of Public Health, as well as the Mosquito and Vector Control Association in the state. So there's a whole network of local agencies at sort of city to county level that do mosquito control in our state. And then California Department of Public Health has both vector-borne disease people, as well as people in their lab in Richmond who are aimed at the viral and rickettsial diseases in humans, doing the lab testing for that. So we sort of view this as a three-legged stool, if you will, partnership to address mosquito-borne disease issues broadly, and Zika happens to be the latest one. So my group hosts the servers that are part of the data flow of this system, and then this map shows you where these agencies are. So it's the dark areas are places with these local agencies across the state. It's pretty much all of the areas with enough people to provide a tax base to support a district to deal with mosquito-borne disease problems as well as pest problems that come with mosquitoes. So from the Bay Area, Central Valley, almost everywhere, Southern California, all the populated areas are covered. Really, I should fill in these areas as well because that's covered by Riverside County Department of Environmental Health. But what this system allows is a lot of data exchange among these different agencies. They can enter field data from their collections across the state, and then our lab at Davis enters the lab results for testing for viruses, for example, and then it all goes into the central system, which updates our interactive maps and then also sends results automatically to CDC. So if you ever look at maps of West Nile, for example, then the data are coming from here to there. We also have the surveillance I mentioned. The California Department of Public Health monitors sentinel chickens for West Nile. This is not what this talk's about, so I'll skip that. But we do have a new RT-PCR test for Zika virus. We weren't too worried about it last year. Suddenly we're having to accelerate our development of that assay. But we extract the RNA and then try to amplify it with RT-PCR, and then ultimately can test for any of these viruses, some of which are our endemic arboviruses. We have dengue, chikungunya, and Zika that are our threats now. So really we're waiting on those of you who are physicians to detect the cases. It's a needle in a haystack if you start testing mosquitoes all over the state without knowing where it is. So I think it's going to be a human case first, then there will be immediate follow-up with testing the mosquitoes that get sent to our lab, and then we may pick it up in mosquitoes if there's local transmission. Now, the big challenge with Zika is the fact that 80% are asymptomatic, So you could get a sequence of cases, all of which could be infectious to mosquitoes, and we actually don't even know exactly how infectious people are yet. But there's evidence it's explosive enough in Brazil that argues people are pretty infectious. So you could have a chain of transmission involving a bunch of asymptomatic cases before you even knew you had a problem, and at that point then you have to initiate vector control. So one of our big limits to the surveillance system is how fast the reporting happens for human cases of disease. And part of it's the asymptomatic problem, part of it's just the reporting system we have to deal with this. And Zika is actually still not on the notifiable disease list. And I know there are very good reasons why that list is limited, but for us that's really frustrating because this, I think it's the Council of State and Territorial Epidemiologists that formulates that, right? And so chikungunya just made it onto the list in 2015 after it had been a problem since late 2013. Zika's not on there yet, so who knows, maybe 2017 it will arrive on the list, maybe well after more cases have occurred in the U.S. So we have a lot of data input structures here. I'll skip most of this, but this is a look at our California Surveillance Gateway. So there are ways to enter your arthropod collection data, your mosquito data, sentinel chickens, there's diagnostic testing modules, dead bird reporting in the case of West Nile, and now we have pesticide application resistance modules as well. We're trying to marry the control and surveillance data. And then Jody Simpson is the programmer that I have who does all the wizardry behind the curtain. We have Google Maps underlying it, so all the sites are registered where they're doing the surveillance across the state, and this will be true in the case of Zika as well. We're also registering the sites that they're monitoring for the Aedes that transmit Zika, so we'll have pretty good baseline data on where the mosquitoes are across the state. They can enter numbers by different collection methods, exactly where they are in terms of latitude and longitude and a variety of other tools. And then we have this is just an RT-PCR plate, but the ways the lab can populate those plates and then indicate the positives for the various viruses we test for. So with the risk for Zika, one thing in California that we're worried about is, well, where exactly are the mosquitoes? And so this is a paper from Moritz Kramer, and I was sort of the California part of this paper, but we didn't have as many dots actually when we did this publication. But this is the global distribution of Aedes aegypti, and this is the dots from a huge literature search, and Moritz is at Oxford and Simon Hayes' group, and they have a wonderful algorithm for searching the web and essentially our scientific literature to scour for all of these kinds of data points over space and time. And then we put them on a map, and then Moritz applies his ecological niche models to provide suitability surface. So that's a very data-driven process. There's a bunch of climate variables and other factors, urbanization variables that then get applied to this, and then statistically what you get is this kind of map. So we have a global map. You might have seen this in the media, even because it's been showing up constantly as the map of Aedes aegypti globally. It's a pretty good way to look at it. What you have essentially is a probability of presence globally, from 0 indicating couldn't possibly be here to 1 indicating it's certain to be there. So this is driven by the data that has all these underlying surfaces informing it so that you get a global map. In the U.S. you can see, but in the U.S. you can see that the strongest suitability at least is in the southeast, and this is pretty consistent with where we do find it. In the slightly less suitable areas that are in pink here, you really don't see the mosquito, at least continuously. And then in California, we have suitability in Southern California and also in our Central Valley. I think there's really not much of a reason why we couldn't see it in Sacramento, for example. And that is a concern. The local district is constantly looking for these mosquitoes, but so far we're limited to areas south of us, like Madera and Fresno. So the data-driven way to estimate suitability is kind of nice. Here's the way the data are collected. These are different trap types. So this is a BG Sentinel trap, and what that has is a lure that sort of simulates a host odors that attract mosquitoes, and so you can run that for a period of time, and then there's a fan in here that sucks the mosquitoes in and then holds those mosquitoes, and they can be tested for virus if you want to test them for virus, or you can leave it out a week and just take the counts to indicate how many you have and sort of track fluctuations in the population over time. This is another major way they're collected, which is simple. It's just a cup. Sorry, pushing buttons here. It's just a cup with a lining of seed germination paper, so it's just a rough surface on which the mosquitoes can lay their eggs. I'll show you what the eggs look like in just a second, but the eggs are a good way to track these mosquitoes because they're not super well attracted to traps that simulate posts, but they do go try and find these containers which simulate sort of what you might have in your backyard flower pots, and then they'll come in and lay eggs on that paper, and then from that paper you can identify what species you have. And then this is a trap designed to kill the mosquitoes, so it can suck the mosquitoes in, and also, I'm sorry, not suck the mosquitoes in. In the bottom you have some foul water that actually attracts the mosquitoes in, so they actually fly through the mesh. And once they get in there, there's this sticky paper that lines the inside, so the female actually gets stuck on there. And the ideal thing about that is that then you're not allowing her to lay eggs and then continue on her way. You're actually killing her, and then you can identify the mosquito and have her not reproduce. So CDC devised this trap, and they've actually provided them free to a number of our mosquito control agencies in California. The one hang-up has been that the sticky papers get dusty and then they're not so sticky anymore. But they're used throughout California. And this is a look directly at our map. If you want to go to it, it's maps.calserve.org/invasive. This is our Google Maps-based look at our underlying data in the system that I house in Davis. And then I put stars on Imperial County because they've been collecting Aedes aegypti as well and finding that they probably have had them for a little while, but hadn't been looking very closely. So Southern California is looking like this, where the blue is the Asian tiger mosquito, which is also a vector of Zika, and the red is Aedes aegypti, and purple is where you find both. So if you go to this address, you can actually click on individual cities and see when they were first and last detected. One little note is that don't assume that's completely complete, in that the districts vary in sort of the timing of data entry, and so if you go there, the information is almost up to date most everywhere, but there are a few gaps like Imperial County. And then I mentioned the trap, so this is what mosquito eggs look like. So one thing we're doing in my group is looking at using the multi-top instrument over in the vet med teaching hospital that they already had for clinical diagnostics of bacterial infections, but we're using that to identify Aedes eggs. So I've been working with Barb Byrne over there, and we're grinding up the eggs essentially and looking at the protein signatures on this mass spec instrument. So what it does, to use lasers to charge and excite the proteins, and then they actually fly through this tube, and so the time of flight has to do with the weight and the charge of the protein. So what you get is a mass spec sort of readout. We got spikes that represent individual mosquito species, and so each species has a characteristic signature. What we've been doing this winter is building a library of species. As you can see, this is 9 different species, actually in Europe in this case, and looking at eggs is not very useful for telling which species you've got. So what they're having to do is bring them back to the lab, rear these mosquitoes, which takes a lot of time and also delays the process, and figure out what mosquito they've got. And a lot of them are native tree hole breeding mosquitoes that they honestly couldn't care less about in most cases. So you want to distinguish those invaders from those as quickly as possible. Just lab testing, this is kind of following on the same things that Dr. Bloomberg mentioned. CDC's tests are all the things he already said. VRDL in Richmond is developing these. It is true the states are behind the CDC, so right now most everything's going to CDC, but VRDL is rapidly trying to develop assays, and all the labs that are dealing with this are sort of overwhelmed because lots of women, as you can imagine, are wanting testing now for Zika. And then these are the tests or the viruses that VRDL can test for. So they have had an encephalitis project in the past, and then they also routinely test for arboviral diseases upon request. West Nile and SLE, St. Louis encephalitis virus, are very closely related, and there are 2 flaviviruses that we've had with us for a while now. And then they're interested in dengue, chikungunya, and then soon Zika as well. Jamestown Canyon is just another one that's pretty rarely tested for. Another thing about using data to estimate where the mosquitoes could be, it's kind of difficult when you're dealing with an invasion. So we're seeing these mosquitoes spreading in California, so if you just take the map and take your climate variables and extrapolate from data, you don't get very useful information about which parts of California are most suitable. So one thing my student Marissa Donnelly is doing is using a reproductive dynamics model where you got eggs, larvae, pupae, adult stages of the mosquitoes, and we're modeling this using math to figure out what the reproductive rate of the mosquito would be, and that involves a bunch of functions that are related to climate variables. And what you get is, in this case, a national map of the reproductive rates of Aedes aegypti. So for the periods of the year that are suitable, what we see is that this is the daily reproductive rate. So what this says is that in the very reddest areas, you get 8 to 10% growth in the mosquito population per day during the part of the year that's suitable. Now, the other part of that is not all of the year suitable. So if you're in Missouri, there's a whole lot of the winter that's not so great for Aedes aegypti. So what we're working out is sort of what are the limiting factors of these mosquitoes and why are they where they are. And another thing we can do then is look at the duration of the Aedes aegypti season, which is the part of the year that is suitable. So in this case, we've got year-round suitability in the very greenest areas, like the very, very tip of Florida, close to the Florida Keys. And then as you move northward, these are the areas that only have about a month that's not very suitable, 2 months, 3 months. And then really where we see the limits of the population is where you get beyond about 3 months, which sort of argues that the cool part of the year is too long to allow the mosquito to get through that part of the year. So she'll be finishing up this work in the coming months, and that work's funded by NASA and happens to relate to Zika just because it's the same mosquitoes. So some of the big gaps are that we really don't know exactly which times are the critical periods for the mosquito-related risk. That's partly because we've got a lot of gaps in our mosquito data. In California, our data are better than a lot of places, and so we have these big holes in our national map where we don't know a lot about what the mosquitoes are doing over periods of the year. We have the maps like Moritz Kramer produces where you got this big global view of suitability, but that views it as a snapshot as if it's that all the time. So what we're really trying to do is move more towards seasonal estimates of suitability. And then we've got just so many trapping methods that trying to make sense of all of that as a big patchwork of data is pretty hard. So guessing what the suitability is requires integrating all of this data in a way that makes sense as a quantity that you can put on risk. And then we don't know much about blood feeding behavior. So I'm trying to get more blood-fed mosquitoes, and we can actually test those to see what the mosquitoes have fed on, but it's pretty hard to find them. The LA Vector Control District, I think, may help me this year and try to get as many as possible. We're thinking about going out into houses, just like we do in Peru, taking aspirators and sucking mosquitoes off the walls of the houses and anywhere they can find them, and just getting as many as they can. Another point about the U.S. is that some of you may know this, but we've also had a dengue outbreak in Hawaii in the past months. And so this is as of January 27th. Here's the timing, December 2015 leading into January. So 241 cases on the Big Island in Hawaii. There's been an outbreak in Hawaii in the past as well. So we know there are parts of the U.S. that are definitely suitable. Puerto Rico is another territory that we often forget about as part of the U.S., but they have dengue a lot of the time, and then Zika is already there as well as being the U.S. Virgin Islands. Another issue with Zika is really the importation risk. So these big circles indicate travelers from Brazil during the period of Zika. So somewhere September 2014 to August 2015, and this is from Cameron Kahn, who's helped us quite a bit with data on chikungunya on flights to and from South America. So in the U.S., you can see we've had over just this 1 year from September to August, 1 year travel from Brazil into the U.S. There was nearly 2.8 million not cases. Don't misconstrue that. But 2.8 million flights into the U.S. from Brazil. And you can see the connections, obviously, to Argentina, Chile, and some of the southern South American places. Even places like Italy, Portugal, France. There's a lot of flights from Brazil. Brazil is a country of 200 million people. It's a major vacation destination. You all know about Carnival, which I think is going on now, right? I think we're right in the middle of it. So Brazil is a major tourist destination, and that's a big issue. As an indicator of what we kind of expect with Zika, this is what we've seen with chikungunya. So this is actually 2015. This is CDC putting 2016 on it, but this is through the tail end of 2015. you can see that in January we get this pulse associated with the South American summer, and then it drops off somewhat in the mid-season, and then there's the summer travel season that also aligns with our own summer that presents risk for Zika transmission here. So in terms of period of risk to the U.S., certainly this is a lot higher due to our mosquitoes and our temperatures. But then we do often see this uptick in imported cases like we're starting to see now. We've had 6 total cases in California to date at last check, and what CDPH has decided to do is to update their data every Friday just so they don't have to answer questions every day about how many importations they're getting. So every Friday the data will be coming out on our importations. And this is for California in particular, and you can still see a little bit of signal and an uptick in January. And then we've got the summer travel season that brings with it more chikungunya. The biggest risk to the U.S. is going to be if it does get strongly into Mexico and starts aligning with our seasons, Puerto Rico, places like that, where truly we'll have cases coming in at the time of the year where we could see local transmission. Another question the media at least keep asking is why now? Why does this Zika outbreak not happen at any time previously? Some part of the story is that the mosquito is just in a lot more places than it was a few decades ago. So the yellow fever eradication campaign, largely fueled by a lot of enthusiasm about some of the pretty harsh insecticides, including oils and Paris green that were used to kill immature mosquitoes, and then a lot of you know about DDT and the problems with DDT, but at that time there was a big push to globally eradicate malaria and let's globally eradicate yellow fever. And so what you can see from that is this is the distribution of Aedes aegypti in the 1930s. By 1970 they'd actually pushed it back in a lot of South America. You can see Brazil is nearly Aegypti free. And then by 1998, this mosquito was back in a lot of these areas, and that's because, of course, the insecticides couldn't last forever in their efficacy, and they had resistance problems, as well as the issues with DDT, with bioaccumulation, and so on. So that allowed this mosquito to re-expand into areas where it had been eradicated at one point. With that has come the expansion of dengue, and a lot of you might have seen dengue trend maps, and over the last 30 or 40 years, there's been this sort of steady rise in dengue. Another question I got yesterday is El Nino causing this? Well, we're all very happy here about El Nino. Of course, it's bringing rainfall to our state, and it brings other things to other parts of the world. The sea surface temperatures over the winter this winter have been about 3 degrees above normal. Here's the graph showing that. So it's on par with the 97-98 event, which was one of the highest ever measured. So far, like 1982, 83, 97, 98, and now have been the last 3 big El Ninos. And with El Nino, I won't go into the details of it, but it's essentially warming of the sea surface along the equatorial Pacific. And this has broad effects on global climate. So this is one of the major engines for our global climate. And what that does, of course, warm sea surface temperatures introduce more water into the atmosphere. You get more storms in that part of the world. So here's the equatorial Pacific. But it also comes with drought in places like Brazil. So the frustrating thing about dengue prediction is that everybody thinks of rainfall and mosquitoes, and it makes sense. If you've got container breeding mosquitoes in backyards, more rain's got to be good. It's not always the case because water storage practices shift dramatically in periods of drought. So actually there can be a lot more backyard water in periods of drought like they're having in Brazil right now. So we don't actually know. It's been frustratingly complicated with dengue that you can't really predict it based on rainfall or lack of rainfall. It's this sort of push and pull, and it's really complicated. So at least the story with Zika is, I would guess, any effect of El Nino is really minor compared to all the other human factors and the other naive population to Zika, and then, as Dr. Bloomberg mentioned, potential interaction with other previous flavivirus infections, and there's just so many things we don't know. But Brazil is in a drought right now, and that has coincided, for whatever that means, with the Zika outbreak. Another thing, just in terms of climate change, which I often get questions about as well, is, well, with climate change, is that, first of all, causing the Zika outbreak, and should we expect Zika in the U.S. and other places that haven't had it before because of it? And this is just an example of a study I've done with CDC, looking at the length of the season for West Nile. And so in this case, we're just mapping the start of the season. I won't go into exactly how we define it, but essentially this is the week of the year when the season starts. So you can see the areas in red are the earliest West Nile season, when it peaks and when it ends. So from that, we can get the length of the West Nile season and then also how high it gets in terms of incidence. There's a strong relationship between how early it starts and how long the season is, and that sort of makes sense. If it starts earlier, it's probably going to go longer. But what gets a little fuzzier, and I think the way we tend to think about mosquito-borne diseases is that we tend to think, well, if it started earlier, we must get more of it, and it would probably reach higher levels. Well, it turns out that's actually not quite true. So an earlier start, which is here, so earlier in the season being on the left, later starts here, versus the maximum incidence, it's a very fuzzy picture. So there's this sort of rough relationship where earlier starts do tend to have a little higher incidence, but there's a huge amount of noise at the national level. And then also the length of the season, same story. Longer seasons, on average, sort of lead to higher incidence of West Nile, but not really. There are places like the Great Plains where clearly the summers are shorter, but then the incidence is the highest we have in the country. Looking at Brazil's case, this is in Recife, which is where the microcephaly epicenter is, and you can see the temperatures there are suitable all the time. Winter in Brazil is not winter by our standards. I think most of you know that, but we're talking variations of a degree or two in terms of mean temperature year-round. We see this in Iquitos, Peru as well. The rains do vary, so there's a wet season, and if anything, the water definitely brings with it dengue as well as presumably Zika as it sort of settles into endemic cycles. And I just wanted to conclude with this phylogeny. So this is from Rob Lanciotti, who's at the CDC in Fort Collins in their vector-borne disease division. And there are three major phylogenetic clades of Zika. And this has been known for a while, but the origins of Zika are thought to be in East Africa. Ultimately got into West Africa, so this is these clades, East Africa, West Africa, and then this Asian clade. And so they have already sequenced the Brazil strain as well as Puerto Rico from December 2015, so that's really recent. They must have sequenced it and then sent the paper off right away. And then Guatemala as well, November and December 2015. All of those are clustering with this French Polynesian strain. So that's where some of the evidence comes from for how the virus went from French Polynesia, perhaps at the World Cup, perhaps at the International Canoe Race, and got into Brazil. It's hard, they'll never know exactly how, but that's the connection. So far we're looking at an Asian strain of Zika virus, and it's unclear whether there's actually been any genetic change that would be consistent with causing microcephaly more readily. There were those hints in the Pacific Ocean, so I think the jury's still out on all of that. And I just wanted to finish by thanking other members of my group and my colleague Art Coffey, who does the virology as the other half of our DART lab on arboviruses, and then mentioned Bill Risen and Bruce Eldridge, who historically also have done a lot of work with the mosquito control and public health agencies in the state and sort of got all of this started. With that, I think that's all I have.
Great. Thank you both. You guys have given us an incredible background on both the disease and the vector biology. I realize it's getting late, but I hope that you'll stay and answer a few questions. Questions from the audience? Yes.
I was wondering if there was any long-term sequela that has become apparent for those who perhaps were infected, maybe found to be asymptomatic later on. If there was any either maybe clinical.
I'm not aware of any long-term sequelae except for, of course, the newborns.
Yeah.
And then the possible association with Guillain-Barré. Not everybody with Guillain-Barré recovers, and so you can have some residual neurologic disability with Guillain-Barré. But that's all I know. The patients with hematospermia, there's been more than one patient with that, and that's resolved also, but that obviously shows a lot of inflammation in the reproductive tract also.
curious if there's any way to estimate this, but it seems like there's got to also be a bit of a reporting bias, right? Because microcephaly was, I'm assuming, wasn't a mandated thing to report before, but now that it's on everyone's radar, it's getting reported more. And has there been any commentary in that from CDC or WHO about how to think about the reporting bias that's likely a piece of this?
That's the reporting bias. I think that's a really key point, because that's the tricky part, is that once you call attention to it, everybody starts reporting that. And we see this in other fields, for example, with vaccines, where people just get a hint of maybe something's related, and so everybody just starts reporting it, but you didn't have baseline data to report. So even with Guillain-Barré syndrome, for example, with one of the pneumococcal conjugate vaccines, there initially was a report of Guillain-Barré syndrome after vaccination. Then you do these large studies where you get background data, and you find out that there's not an increase. Yeah, sure, you can get vaccinated today and next week have Guillain-Barré syndrome, but you could also not get vaccinated today and get Guillain-Barré syndrome. And when you do these very large epidemiologic studies, you can document that there's no increased risk. So that's why there's a lot of caution with talking about these associations, both with microcephaly and Guillain-Barré syndrome. However, with microcephaly, when you isolate the virus from the mom and from the child, that's starting to look positive.
Are they screening presumably for other viruses as well that are linked, CMV?
And that's not known either. So because it's not known, the workup is not standardized.
I was going to ask the same question, as whether or not prior to this starting, whether or not microcephaly was a mandated reporting, and how thorough was that being reported, and leading to the question of report of ascertainment bias here. So is it a mandated reporting, and how robust is the reporting capability in rural Brazil, which is what we're talking about?
Right, no, it has not been mandated. Right now, of course, people are swarming into Brazil to design the studies, so I'm sure it'll get much better.
I was curious if you think it's been chronically underreported for years, because, if you're talking about 25,000 being a normal number for the U.S., you've got a country of 200 million with a normal of 150, then 4,000 doesn't sound so bad. So I'm curious. I'd hate to think that all of this is caused by over-reporting, but definitely seems like it's
Right, so that's the thing. It's really the magnitude of the risk. What is it? Is this a small bump, or is it a huge bump? If you look at the data, the numbers that are reported, it makes it appear as if it's a very large bump. But for all we know, it's only a 1% increase or something. And there's just once you get started and saying there's association, everybody starts reporting it. And a lot of these kids with microcephaly have been investigated, and there has been no evidence of Zika virus infection.
I did see today on PBS's website, which I consider to be somewhat reliable, they had a story of 12 infants that they tested cerebrospinal fluid, presumably for IgM. Would that be right? It was for antibodies, and they didn't say which, but they found all 12 had evidence of Zika infection. And they all had microcephaly, and they're working back to test for Zika, and all of those 12 had Zika antibodies. So 12.
But he suggested, right?
Yeah.
So I'm just doing the trial clinic, so I'm kind of interesting. So in my understanding, the antibody of Zika virus can have some cross reactivity with dengue antibody. So it has cross reactivity with other flaviviruses, like yellow fever, etc.
Do you want to take that?
No, please.
So the flaviviruses, there's actually more flaviviruses even than dengue, yellow fever, and Zika down there. Chikungunya is an alpha virus, so that doesn't have a cross-reactivity problem. But clinically, they look the same. But in terms of serology, really with flaviviruses, what they're looking for, if they want to see whether you were infected with flavivirus serologically, they're going to test for IgM in the acute sample and then IgM in the subsequent date, and look for a rise in antibody titer. And feel free to correct anything I say that's wrong. So, and then also for flaviviruses, they'll compare against reactivity to other flaviviruses. So as I said, you give reactivity to dengue if you had a Zika infection, but they look for a fourfold difference, so you should be more reactive to the one you got. And where it gets complicated is if you get sort of a third flavivirus infection, then it's a mess. And so for dengue, we deal with that all the time. We work with the U.S. Navy for our data streams. They have febrile surveillance in Peru, for example. And if you get 3 serotypes of dengue, then we don't know what you got. You got dengue is all we know. But we don't know which serotype. And that's notorious in dengue in particular, but then Zika is going to throw more wrinkle into that. Rocio is another flavivirus that's transmitted in Brazil, actually. Not really been noteworthy, and I assume serologically there's going to be some issues coming from that as well. Another mosquito-borne flavivirus.
So the PCR, how long PCR can be posted?
PCR is not long. I mean, it's maybe up to a week, but it's...
A week is that's what I've heard, is about a week.
Yeah, so depends on one thing. How long should a couple avoid some sexual activity? One partner returning from some endemic area.
See, that's not known either. So, there's just been a few studies detecting Zika virus in the semen, but we know, for example, with Ebola virus, that patients who recovered from Ebola sometimes could excrete Ebola virus in the semen for months afterwards. So I don't know if that might serve as a privileged site of infection and reservoir.
Actually, yeah. When they first started talking about Zika, they were saying that there was a possibility that these very bad reactions were possibly as a cause of a previous dengue, and then have Zika either on top or after it. Is that still being looked at, or has that been discarded?
Well, so I think the origins of that are largely in sequential dengue infections. So with dengue, if you get that second infection with a different serotype, there's what they call antibody-dependent enhancement. And so the mechanisms are sort of harder to work out, but we know that severe dengue is more likely if you've had another serotype of dengue previously. And so I think that's where the hypothesis comes from. I don't know if you know any clinical evidence on that, but I don't think anyone's really done anything with Zika to know that.
No, and if you think about it theoretically, I mean, there is that theoretical reason for that to happen with dengue, but that's disease severity. And when I think about the fetus being involved and the microcephaly and all, I think of other things like cytomegalovirus and rubella, and those don't depend on severity of disease. And rubella is a very mild disease. There's no reason to have a vaccine against rubella because of the disease itself. People get sick, they get better. It's the sequela, it's the intrauterine infection. So to me, that doesn't make as much sense. But whether that might, but then again, there might be some that might serve as a cofactor in a way that I'm not creative enough to think about.
Yeah, we've heard a lot of some of the resource-poor countries, obviously in Central and South America, are extremely concerned about this apparent epidemic. And I believe that I heard that in El Salvador that the National Public Health Department had actually recommended that the entire population of the country refrain from becoming pregnant for 2 years. I'm not aware of any precedent in the history of international public health for such a recommendation. Is that something that has been discussed internationally? And any thoughts about what the effectiveness of such a recommendation might be?
Well, I've thought about it. I like kids, so I wouldn't make that recommendation. But pregnancy is a very personal decision, right? There's a window of opportunity to become pregnant. Maybe waiting 2 years means you're not going to have children. And this 2 years, as far as I can tell, the idea was that since everybody's susceptible, that this infection is going to go through the population like wildfire, and within 2 years everybody's going to be immune. I don't know that that's going to happen, though. So I think that's a bit speculative to make that kind of recommendation. And it's a very personal decision. So people should consider that, but I don't know that I'd make the recommendation not to get pregnant. That seems too personal.
As an adolescent doctor who spent most of my life convincing people not to get pregnant, not very effective.
Or to try that across the country.
I would recommend And maybe people will follow it.
Maybe one more question, yes.
From a medical and public health perspective, what are the risks for, say, a 20-ish non-pregnant woman traveling to Brazil this summer? Like, will there be a possible risk of, say, having kids in the next 5 years and that kid having microcephaly?
As far as I can tell, there's no evidence that this virus is, for example, similar to cytomegalovirus, that once you're infected for life, and that it could reactivate and cause problems down the line. I'm not aware of that happening with this virus. So all I'm aware of is if you get infected during pregnancy, and I would hypothesize probably during the first trimester, although there's no data on that, that's the only risk period. So that traveling now, the worst that could happen is you would get Zika virus infection, you'd be mildly ill for a week, and you'd be immune, and when you came back, you couldn't donate blood for 28 days after you return. That's my best answer to that.
We'll have to stay tuned. We'll keep you guys posted. Thanks again to our two guests, Chris and Dean, for coming on and spending the evening with us. Thank you for your time, and we'll hopefully see some of you again next month when we have our next Global Health Forum. Thank you. How are you doing?
He's been on the program, so I asked for the job. Oh, no, you told me he's in the program.
You guys are in the hospital.
Yeah, we're trying to get them off.
Now's the perfect time.
Your family and I are working.
No.
No, all right. My doctor's there all the I've been really an example of the healthcare.
First thing to do.
You guys have okay, I'll be in touch. We'll get together sometime. Thanks so much for
That's what I'm saying. Yeah. They are awesome. I know, right? All right. My first teacher back. We haven't learned that yet, guys. That's okay. I know. Right? It's like what it tastes like. What are we doing the rest of it?
Take it home. I'm taking one home.
Thanks so much. I'm so sorry about all the chaos. It sounds to me like
Everything's done. Yeah.
Blair.
Okay.
So he tested this, and then I didn't know what he needed to do.
Right. Oh, don't worry about it. I know it. Yeah. Keep in mind. Yeah. Okay. And everything got fixed in the gardener showed up too.
Well, I suppose
That was fixed too. I know.
He said he didn't. I talked to him because I never worked for him. I said, you've worked for years.
Yeah.
So I had to send him a Google map, and then finally he got his dog.
Okay. Yeah. Thank you.
But
we brought over like. Okay.
Thank you, Dr. Borg. Hello, nice to meet you. I'm Leanne from the ID Field.
Oh, hi.
So I'd like to welcome you at this conference.
Where are you from?
UC Davis.
No, you speak with your accent.
Oh yeah, from Japan.
Japan.
Sorry, yeah.
Okay, yes, just got the information. We have some students who just arrived from China and a couple of others from South Korea. Anyways, thank you very much for coming.
Thank you. So I'm actually new at this conference. So I'm just trying in the future, hopefully get the.
Very cool.
I know.
We'd love to. If you email me.
I'd love to, do the plug today and everything,
That's how we like to do it. Clean out your phone. Bring it over here.
Sure, very cool. Thank you so much.
I look forward to seeing you. Thanks for your question.
I know.
So yeah, I will definitely do that.
Yeah.
How are you doing otherwise?
Very good, actually. I'm very good.
What's the PI? I don't know because so you're not working by your phone.
A little wild today.
It's a Thursday night.
Good.
Trying to correct one of the locker.
Well, but it's great. Yeah.
Thursday? Or you're not
And I'll keep thinking of you. I'll keep trying to find opportunities for you.
Thank you for thinking about me.
I appreciate it. Yeah, and I projects with Julie ideas together. If any of them gel, we'll let you know. Okay, cool. Thanks so much.
Look at other.
Yes.
Aluminum.
For recycling.
I don't know if we're supposed to take care of this stuff. Hillary didn't
Well,
want
you
to
can
talk.
no, it would be in a hangar.
You guys, all in my background.
Give us as much notice as you can. Yeah. You got 14 minutes.
There's enough of us.
I'll take. You want to put that on?
Vegetarian burritos?
No, but I am a vegetarian. No, I've got
We say two just for you.
I'll have to do these for you. Thanks, Tom.
Alright. I'm gonna do this. Okay, yeah?
Calvin, you should use your helmet as a bucket. Oh my god, you guys, can one of you bring two of those?
I'll bring stuff too.
Okay, never mind. That's the carrying. Yeah. Shane, are you taking these home? Set them in your backpack?
Gym lab figure.
I can do it. I got it.
With rubella, it can carry that. And for example, with HIV transmission and hurricane, but that accounts for about 1% of carrying
I'm carrying
HIV transmission. The vast majority occurs during the birth process.
Right, yeah,
itself.
that's what I was under the assumption. So,
Right.